Hello, my name is Stephan Urban. I'm a professor at the University of Heidelberg in the Department of Infectious Diseases at the Molecular Virology. It's a pleasure for me to introduce you today into hepatitis D and hepatitis delta virus. Let's start with the ABC of hepatitis viruses. Hepatitis viruses preferentially replicate in the liver, where they cause inflammation, which we call hepatitis. Some of them, the hepatitis A and E virus, enter via the gastrointestinal tract. Others, B, C, and D, are transmitted via blood contacts, for example, by small injuries and wounds. Now, in contrast to A and E, the hepatitis B, C, and D virus can cause a chronic infection that has a progressive inflammation. Now, let's have a look at these 5 viruses. As you can see in that picture, they are different in sizes, they are different in their shape. Some of them are envelope proteins, the B, C, and D virus. 2 of them are naked, and some of them contain RNA, and the hepatitis B virus contains DNA. Now let's focus on the special relation between the hepatitis B and the hepatitis delta virus. Let's start with the delta virus. It's a satellite of hepatitis B virus. That means it requires hepatitis B virus proteins in the same cell where it replicates. And the genome of the delta virus is an RNA, and that RNA is packaged with delta antigens that wrap around that RNA. And this ribonucleoprotein complex, as we call in the mature particle, is enveloped by proteins that are not encoded in the RNA, but they are encoded in the DNA of hepatitis B virus. And this very special relation, the dependence of envelope protein from the B virus, characterizes this kind of symbiosis of these 2 particles. The D virus is the smallest human pathogen that we know. We have about 260 million people that are chronically infected with hepatitis B, and they provide the replication space, as we can say, for the delta. More than 12 million of those patients or individuals are co-infected with the delta virus. And this chronic delta co-infection causes the most severe form of viral hepatitis with a faster progression of an infection, of a chronic infection, to cirrhosis and at the end hepatocellular carcinoma. We don't know the real numbers of the co-infected patients due to the lack of epidemiological data in many countries. We know Mongolia, Africa, South America have quite a high incidence of hepatitis delta virus, but we need better data to do that. And we have a difference. We have very effective drugs for chronic hepatitis B virus infection. These drugs do not directly affect hepatitis delta, and we have only one drug that's approved for chronic hepatitis delta, the entry inhibitor bulevirtide. Now, the transmission of delta is sexual transmission, contaminated needles, tattooing, or direct sore contacts. It's rarer than for hepatitis B alone that a mother-to-child transmission occurs. And we distinguish 2 different types of infection. The one is the co-infection, a co-incidental infection with both virus from a co-infected patient. And most adults that have this kind of co-infection can resolve an infection by the immune system. However, if an already existing individual with chronic hepatitis B gets a super infection with the hepatitis delta virus, there's a high risk of developing a chronic infection with HDV. So these patients or these people are at high risk of acquiring a delta super infection that is continuing in a chronic way. Now, consequently, an HPV vaccination prevents from an HDV-HBV co-infection because both have the same envelope proteins, and the antibodies that are directed against the envelope proteins neutralize an infection. However, an HPV positive individual should know its HDV status in order to be aware whether it has already a super infection, and it's of course in the risk of getting a super infection. Let's have a very short look at the replication cycle to understand how we can interfere with hepatitis delta virus replication. Now, the D and the B virus recognize and enter hepatocyte via a bile acid transporter. We call that NTCP. And this step is the step that can be blocked by bulevirtide, an entry inhibitor that exactly binds to the site on the receptor that is required for the virus to enter. Once the virus has entered the cell, it's transported to the nucleus and replicates by a quite complex mechanism that I don't want to go into detail. But unlike HBV, there is no HDV-specific enzyme required for replication. The virus is doing that with host enzymes. And the consequence out of that is that the nucleoside analogs that are inhibiting HBV replication cannot block HDV replication. Now, the HBV envelopes that are then required to form the mature particle are provided by the cccDNA from the hepatitis B virus in a co-infected cell, or they can be provided also from an integrate that occasionally occurs in an HBV infection, and that leads to the release of progeny viral particles. This way of spreading leads to an amplification of the virus within the liver and between individuals by a co-infected cell. So virus comes in, virus comes out, can infect the next cell, can amplify if HBV is there, and can amplify further. It can also infect a non-HBV co-infected cell. And this process is blocked by an entry inhibitor like bulevirtide, but also by envelope-specific, HBV envelope-specific antibodies. But we know that there is a second way of replication of that virus, and it's important to understand that for a possible curative treatment. And that is that if such an infected cell divides, it can propagate the viral RNA to the daughter cells, and both of these cells may then inherit HDV positivity. This is an HBV independent process that may occur in the liver, but this is inhibited by interferon. And if we block these both spreading pathways, we have good chances in order to eliminate that virus. And you will learn more of that from the further lectures where we have the therapeutic effects. Let me just end up with that summary of possible spreading mechanism of the virus and sum up with some take-home messages. So chronic hepatitis D is a rare, underdiagnosed progressive liver disease that manifests only on the background of an HBV infection. HBV positive individuals should be tested for Delta and are at risk of superinfection with Delta via blood contacts. The Delta virus requires the HBV envelope proteins to form particles, but its RNA genome replicates independent from HBV. That means that replication is insensitive to nuke treatment that are active against HBV. So we need different ways of treating. And it persists via particles in body fluids enveloped in HBV, or via transmission of genomes from cell to cell during division of hepatocytes. If we control the de novo infection of hepatocytes by an entry inhibitor, we can control the infection if we do that continuously. And we may eliminate HDV from infected cells by a durable block of entry, and we would have to inhibit cell-to-cell spread, possibly by interferon or other novel drugs. With that insights, I would like to end and thank you for your attention.